this is my title. And up front, let me say, I will try to keep technical details to a minimum. We have plenty of time to discuss details of methods and stuff like that. I would rather show you examples of what you can do with these methods. But in some cases, I'll explain a little bit as well. I would like to start with this one. Just to remind ourselves that microbes have actually evolved to operate in rather complex conditions. I call this a fitness landscape. It could be many things. It's just not just the physical landscape, the habitat. It's also the resource landscape. It's predators. It's competitors. It's colleagues, if you mean. So all this is a part of the fitness landscape. And that also means that microbes are pretty good at responding to changes in context, right? Context. You move, for instance, these guys out of the natural habitat into a petri dish. This is a massive change of context, right? And they adapt to it. Or you want to culture something from a habitat, you culture what you ask for. So there is a lot of information that about the natural environment, the natural context, the fitness landscape that we often are lacking. So because we largely study microbes in vitro, right? And then we, from that, infer things about what's happening in vivo. Also, for many good reasons, we study models in the lab, where we can do it at very detailed cellular level, typically. And we then try to infer from these kind of mechanisms and interactions what is going on in natural communities. But often, this work is not well informed because we simply don't know very much about what's the natural habitat, what's the context of the microbes. And this is because we still need better in vivo measurements that can tell us that and inspire us to get better models, for instance, or to interpret some of our in vitro measurements. And I also say it's also important to consider scale, relevant scale. I don't know what relevant scale is. We can discuss this. It's an interesting discussion. But it's more important to be aware of what scale are you measuring, because you make a choice, and that gives you also already some of the answers. So if you're interested in the system, the dog, how that responds, put the dog in a plastic bag, measure how it consumes oxygen, have a fine measurement of the system, but it doesn't necessarily tell you what's actually going on, who's involved. Or if we do the opposite, we take a little sensor, we see sensors in a moment, we measure at the scale of the microbes. We get a lot of information about the microbes and what's going on, but it's not easy to make the extrapolation up to the system level or some of the emergent properties that are coming out of these interactions between the microbes and the system. So scale is important. So in my talk, I will focus on two things. I'll give you an example of microscale measurements, but then also show you some techniques that enable you to do imaging. They can start to sort of move away from point measurements to more spatial measurements. So one thing you can do, there's a great tool set around, is using miniature sensors, microsensors. They've been around for many years now, pioneered by Niels Peter Røsbæk at the University of Aarhus, who introduced this concept in microbial ecology. And if you look over the list, we can measure many relevant things, right? Compounds that are involved in microbial metabolism. And we have different measuring principles. We have electrochemical measuring principles, and we have also optical measuring principles. So we can also measure light. We can measure also chemical parameters using optical sensors. And so these are very nice tools. You can get highly specific information at high spatial resolution about what's happening where you're measuring. And that's also a limitation, because they're point measurements in a three-dimensional, or should I say maybe four-dimensional world. So, but you get very many informations about gradients and things like that. But it's a one-dimensional approach. You can also use some of these measuring principles to go beyond this. And there we go to the optical chemical sensors. So an optical chemical sensor is a sensor that relies on an optical change in an indicator, a reversible change. Here it's shown for an optical oxygen indicator. So you have a dye, you give it a pulse of excitation light, it gets excited, and after a while it gets de-excited and emits phosphorescence. Now, if this happens when there is oxygen in the vicinity, oxygen can collide with the excited indicator, take over energy, and that then dissipates via singlet oxygen. And then you have less or no light emission. So you can easily measure oxygen by measuring the amount of red light that is emitted from your sample with an optical sensor principle. So the nice thing about this, there is no net oxygen consumption in the measurement. So that means you can scale up your measuring volume or your sensor area relative to your sample, and you do not affect the real oxygen dynamics in the sample. And that means you have many different possibilities to make and use optical sensors. So you can, of course, make an optical oxygen microsensor with comparable measuring properties as an oxygen microelectrode. But you can also scale it up. We put a sensor patch inside a volume. But in this context of the talk, you can also put it on a foil, or you can put it on sensor particles that you can then use or employ for imaging. And we will look at some examples later on. And there's many systems I could show you, biofilms, sediments of different kinds, but I decided to show you a different system. I'll try to take you on a journey through one particular system where we employ these techniques to really dissect the different compartments and what that tells us about the living conditions for microbes. And this is the Kuehl symbiosis. So Kuehls are these small calcifying polyp-animals that form clonal colonies up to several meters across. And they're really the builders of the coral reefs. They are polyp animals. They can catch prey with their tentacles, stinging cells. They do that for nutrition, nitrogen, phosphorus. But actually, their carbon demand is covered by a symbiosis with microalgae that sit in their endoderm tissue here and then can cover 80 to 90% of their carbon demand. And that's key to the Kuehl symbiosis. So there is a symbiosis with microalgae, but just as important, there are also lots of other microbes, bacteria, archaea, virus, fungi, spread over the Kuehl colony. And therefore, we also talk about a multi-species assemblage, a holobiont here. So we're looking at a holobiont, like we also are holobionts, right? And so what can we learn about the living conditions in such a system with these different techniques I talked about? So I'll first show you some examples of microsensor measurements, temperature, oxygen, pH, and we'll go by compartment. We'll start the outer tissue layer, which is covered by mucus. As water flows over the tissue, it decreases in flow speed, and that creates boundary layers. That creates mass transfer impedance. It slows down mass transfer. And that means that you very easily create gradients. So if you look here at the water, we have the cold tissue. In light, there's oxygen produced by the microalgae, but it has a hard time leaving because of the diffusive boundary layer that slows down the export. So oxygen builds up to very high supersaturating levels here. Reversely, in the dark, the only way oxygen can reach the core is across this diffuse boundary layer. So therefore, the core tissue gets hypoxic or even anoxic. And that's very important, of course, for the core to adapt to. Likewise, pH is affected by photosynthetic activity, high pH in light, low pH in the dark. And this is strongly light-driven. And if you do cycles between light off, light on, you can see we can go from literally anoxic to hyperoxic within 5 minutes. An extremely dynamic environment. pH shifts several units, a unit or two, within, say, 10, 15 minutes. And that's the reality. That's the reality of microbes, not only in this system, in many other systems.
So would you be willing to say a little bit more about what it is exactly that allows Henry's law equilibrium to not apply in the depths of this?
Which kind of equilibrium?
Henry's law, like the fact that the solubility of oxygen in water is usually order 210 micromolar.
Yes, but it's because there's consumption. There's respiration here, and there's production, right?
I see.
So it's not just water. Okay, is that okay? Also, corals are pigmented colonies. They absorb solar radiation. That means they heat up. And again, boundary layers, there's a thermal boundary layer. That means that actually on average, a coral in an illuminated situation is actually warmer than the surrounding waters. And you can measure that as well. You can see here, the lower the flow, the higher the temperature at the surface, so the thicker the boundary layer. Or here, the more energy you hit the corals with, the higher the temperature. Right? There's also light gradients. So pigmented animals, we can use small sensors to quantify the total amount of light in the tissue, at the tissue surface, in the tissue. And what you see there is that the light field is very different from the incident. So this is here showing you the total flux of light, different wavelengths in percent of the incident light. And what you see is that the spectrum shows dips. So it's modified by the presence of pigments. But you also see that close to the surface, we have enhancement of light. And this is due to very strong scattering in the tissue and from the skeleton. Again, a very different reality from the incident. And often we think about these systems as passive exchangers of temperature, of heat, or mass, or oxygen. But actually, the surface of these corals are covered by cilia. Like our airways, for instance, are covered by cilia. And they beat in synchrony, and that has consequences for the mass transfer in these systems. Because the cilia movements induce vortices at the tissue-water interface. And that enhances advective flow. So to the extent that the corals can regulate the cilia activity, and we're looking into that at the moment, they can actually affect the exchange in a more active way than just relying on passive diffusion. So how to study this? What we do is we do particle image velocimetry. We seed particles, fluorescent particles that flow over the coral, and then you can see here that while it flows in this direction up here, as you go close, you can see how particles rotate around, and those are the vortices. This is just a zoom, right? So we recently published a system where we can do this in 2D and 3D. It's all open access. You can read the details. I can tell you about the details afterwards. So we can really align flow patterns to surface area. For instance, how does the flow over an expanded coral colony compare to the flow over a contracted coral colony? What we can also do, we can expand this measurement by simultaneous measurements with oxygen. So how does this work? It actually works in the way that you use flow tracking particles that are also reacting to oxygen. And to make this work efficiently, we recently developed a method so we can measure oxygen via this phosphorescence decay. It's a lifetime proportional imaging method. We can do this very fast, so we can measure oxygen very fast, and we can at the same time track the flow. Just to illustrate how this works, try to look at this. What we have here is a cuvette. It's filled with oxygen sensor nanoparticles. Here we are dripping crystals of sodium bicyanate, which is an oxygen scavenger. It reacts with oxygen, removes it. Here you see an image of the resulting oxygen concentrations as the particles sink down. You can see how they scavenge oxygen as they sink down. And you can see here at the same time, average flow field. So we can really combine flow and oxygen in very dynamic settings, yes?
How big are the particles?
In this case, the particles are 200 to 400 nanometers.
Do you worry about like quenching via interactions with molecular species that aren't oxygen?
They are sulfur dioxide is a known quencher, but that's the main one I would be worried about, and you don't find that normally. Okay, so let's put this in a call context. Just an example of what you can do. So what we did here, we basically did measurements of the cilia beating frequency as a function of increasing temperature. And you see, relative to the control, you see how the beating frequency goes up to a threshold. And then the call basically collapses, the cilia beating collapses. And that has consequences for the mass transfer on the call. So you see here on the left, this is the oxygen, 2D over the call surface. You see how there is a concentration boundary layer here. Actually, what happens in the dark, so it's consuming oxygen, with the color scale, so the darker, the less oxygen. But as it increases frequency of beating, you can see the extent of the concentration boundary layer increases, and that also drives stronger deoxygenation. And when it collapses, see here, cilia beating more or less stops. It collapses to a simple diffusive boundary layer here, and literally at 39 degrees, the core dies. So this cilia movement and the advection induced by it seems to be extremely important for this system.
How big are the organisms again? In this setting?
I think this here is something like 5 millimeters. Zoom in, I think, something like that. Okay, so I move to another compartment now. That's the stomach of the call, which is an extremely important compartment. We don't know very much about it. Anything that goes in needs to come out. It don't have a gut like we have. So it's important for particle uptake, pre-digestion by microbes, generating essential molecules. It can also be used for excretion of waste products, particulate waste products. And actually also the symbionts have to be recruited via the digestive cavity here. And so what's going on in there? So if we stick a microsensor in this, we'll see that again, we're talking about a very dynamic microenvironment. The stomach is largely anoxic in the dark. At light, it's oxic or even hyperoxic. It's affected by the surrounding tissues. pH decreases with depth, so we have often gradients in the stomach. And this is, of course, also interesting from a microbiological perspective. So there is quite some niche space here for microaerophilic or anaerobic microbes to participate in the digestion of particles. But this is not a steady state situation. Actually, if you leave a microsensor overnight, you see some calls are able to keep oxygen around atmospheric saturation, close to 200 here, while others struggle, but it's not constant. And that has to do with host behavior, because host behavior also plays an important role here. Look at this. This is an example of one of my postdocs. Put a microsensor in and just monitored with a time lapse the movement of tissue. And this is the oxygen down in the stomach. And you can see the behavior of the call is able to modify the conditions in there. Okay, alcohol is rising, so I speed up. But this was a microsensor measurement, but actually these polyps are connected. So they can share water content across the polyps. So So can we actually image what's going on there? Yes, we can. We have developed a technique where we can inject these nanoparticles inside the gastric cavity and follow the distribution between polyps, and actually also measuring the oxygen. I'm speeding a little bit quickly over it here. So what we can do is, here we have a coral piece. It's about a square centimeter, injected with particles. The red one, 10 minutes after, they stay in the system for 24 hours. And what you see here is a live recording, basically, of oxygen in the gastrovascular system over time. And you see how it looks like a kind of a calcium imaging of neurons firing. You see how this anoxic water is being moving around, and it's strongly affected by the behavior. So microbes are also affected by host behavior to a large extent. Okay, very quickly, the last compartment, I won't say much about it because it was planned to have in the first methods talk, but didn't get to it. But anyway, the last interesting compartment here is the skeleton compartment. It's actually the largest compartment on reefs, but it's a compartment we know the least of. And it's a highly shaded environment. There are algae living there, but on a very low light, we're talking 1% or less than 1% of surface light reaching this layer. And also there, we can image the oxygen conditions. In this case now, not with particles, but with a foil. We make a cross-cut, we put our coal on top of that foil, and that tells us the distribution of oxygen. We can also get structure at the same time, and then this is an overlay. So this is another example of techniques you can apply on a natural system. Okay. So let me just two more slides, then I'm done. So I think what I wanted to communicate is it is important to account for microniches and the spatio-temporal heterogeneity when we sample holobionts or any natural microbial community. So there are different characteristic compartments. So if you look here on the coral, there's a spatially structured landscape with different microbial inhabitants in different compartments. And they operate under different microenvironmental conditions. Some of these inhabitants are in contact with each other. They maybe move into other compartments, but other microbes maybe just indirectly sense the result of metabolism somewhere else. And some might not ever see or feel each other. And that's an important message as well, because if we sample at larger scale, at too large scale, if we lump the whole holobiont, extract all of the DNA out of it, and then we start to make correlations. We can make correlations, but do these correlations reflect reality? We don't know, because we don't know the underlying structure. So another message here is I think we need to align sample size and analysis with the underlying spatial structure and microenvironment of such holobionts, or for that sake, any other microbial community you study. And that I would like to thank my nice group and also some key collaborators, including Doc, who's in the audience, and then also, of course, the funding agencies. Thank you very much.
Thank you, Michael. That was great. Time for a few questions. We're trying to allow junior members of the group to ask those questions, if there is any. Okay, not often.
Great talk and great science. I want to sort of invite you to speculate, and maybe you have data on that, but so all these fine resolution concentrations of chemicals, gradients, fluctuations that you measure, do you have any sense for how much the organisms are actually in equilibrium, metabolic equilibrium with what you see?
Yeah, it's a good point, actually. Often when we do microsensor measurements, we go for steady state, right? We do these profiles. So we change the environment and we wait for a steady state. Profile doesn't change anymore. We assume steady state between transport, consumption, and production. But that doesn't necessarily reflect the reality, right? On a reef or in many other natural contexts. So, but I think it also depends on what you're looking at. So if you're looking at a small volume like the Kuehl stomach, which is only a few microliters, I think you can assume good equilibrium. But if you're looking at, for instance, how the tissue responds to changes overlying water or in the gastric cavity, this just by sheer distance calculation takes time. And but I think it's a very important point to not forget about the temporal variability in the resource landscape. I don't know if that answered my question. Your questions, yeah.
So we theorists are often lazy, so we like to just assume that environments are random. We pick them from a Gaussian distribution or something. So if you were to put all of your measurements in a bag, and if I pull them out randomly, would I get a Gaussian?
Oh no, I'm not imagining.
You have all the numbers.
Yeah, give it a try.
I think there was a question maybe at the back. No, all right, Benny.
So about sensors, how sensitive and how specific are these sensors, and what is involved in developing a sensor, right? For some, for some compound you're interested in.
Yeah, that's also a good question. It's also a long task, a long
I know
journey.
exactly.
Generally, when I talk about a sensor, that's actually another point. So there's indicators and there's sensors. If you look at the microscopy field, a lot of things are called sensors, which are indicators. They react with the compound and change the fluorescence, but they're not reversible.
Oh.
So a true sensor is something which is, in my universe, specific and reversible, and in the best of all worlds, also fast responding. And then when it comes to how do you develop a sensor, if you're interested in electrochemical sensors, it's good to look in electrochemical journals. And there's a lot of there's a lot of good electrochemistry done for centuries. Right?
Oh.
But what you can do is, yeah, but if you have a sensor principle, that's the start, right? So you need a material that you charge, for instance, with a potential, and then it reacts with a compound. But to make it work as a microsensor, you need to scale it down, because you're not interested in a sensor that consumes a lot of analytes and makes its own microenvironment, right? You want to probe the real microenvironment. And that's where the geometry issues, the handcraft basically of making small glass sensors and so on come into play. But I mean, in many cases, these electrochemical sensors you saw on the list are actually, by measuring principle, electrochemical measuring principle, are analog to large electrochemical sensors, which have been scaled down in a clever way.
All right. One last question, perhaps.
So I just have a question, like say, have you guys applied these kind of sensors to plant leaves?
Yeah.
Do they look qualitatively very different? Or
Yes.
actually...
If you mean terrestrial plant leaves, yes. And that's because oxygen diffuses 10,000 times faster in air than in water. It's the same if you use these sensors in soil, unless they're waterlogged. You'd never get these nice profiles. But if you water saturate the soil, you can. Yes, we have used them in and on seagrass leaves, for instance, that was beautiful, or rhizospheres. Oh yeah. But as soon as you have a mixed air-water system, then of course this diffusion thing starts to.
So the diurnal, do you see the diurnal cycle very?
You get huge variations. You measure a soil profile. So, yeah.
All right, let's thank Michael again.